Maybe we will have the presentation in English today. And I have the very immense pleasure to introduce Dr. Maria Adelaida Gómez. It's a good friend. There was a friend, one of these online friends that you talk to people online and then you get to know in person, and she's really a lovely person. We were at the Chagas disease meeting at Caxambu, and she came from Colombia for the meeting, and then she gladly accepted to come to visit us at CTVacinas. And Adelaida, I need to read, she has a PhD in microbiology and immunology. She's from Colombia, and she got a PhD in microbiology and immunology at the McGill University in Canada. And then she's now the head of the molecular biology and biochemistry lab at the Centro Internacional de Investigaciones Medicas in Cali, in Colombia. And she's one of the great experts on human leishmaniasis. She does very nice work that's going to show to us today on the immune response of patients with cutaneous leishmaniasis, but also we were working together on visceral leishmaniasis. And I also would like to acknowledge the presence of Professor Selma Jerônimo. So we have the two maybe biggest experts in leishmaniasis here in South America, I could say easily. Professor Selma Jerônimo is the director of the Centro de Medicina Tropical at the Universidade Federal do Rio Grande do Norte. So, and she's also a good friend and collaborator in the Leishmania vaccine project. So you all know Professor Selma. Thank you, Selma, for coming here too. And so I'm very happy that you're able to come. Thank you again. The phone is yours.
Thank you, Santuza. For me, it's a very immense pleasure to be here. It's the first time that I am in Belo Horizonte, and unfortunately I'm going to be too short, but it has been beautiful. Just this scenery behind our meeting room is just amazing. And Santuza, thank you so much for the invitation. I'm very happy to be here. So as Santuza was saying, I come from Colombia. I have been doing research in leishmaniasis for about 20 years. And what I want to show you today is a little bit of the insights that we have gotten in the past 10 years or so on the role of the immune response in the outcome of treatment of cutaneous leishmaniasis. And this, as I put here, this conundrum of having a very inflamed lesion with very few parasites. How do these coexist? So before I start, I just wanted to give you a very brief view of what CIDEIM is. So we are a biomedical research center in Cali, Colombia. Of course, you all know where we are, but we're in the South Pacific area of the country. And we conduct research on neglected infectious diseases, and we have a line among the different infectious diseases that we work with on cutaneous leishmaniasis. So CIDEIM has been conducting research on CL for about 60 years now, and we were for about 40 years or so a WHO reference center for cutaneous leishmaniasis for the region. And we have been doing research all through the different spectrum of essentially possibilities that you can get, from very basic research to applied research. And in the past years, we have been involved much more in community-based research to try and get the community involved in decision-making towards what are the research projects that are important for the community themselves. We are also, and I bring this slide is because this may be of interest to you, we're also a regional training center for the TDR. So that means that we are a hub that hosts a plethora of different online courses and events that are devoted to promote capacity building in research. So we provide a lot of courses on project planning and evaluation, on biostatistics for biomedical research, on implementation research, and this type of things that are promoted by TDR. So if you're ever interested in those courses, just go to our webpage and you will find a lot of resources that are free to access and that would be useful for you as well. All right, so going into detail, I didn't bring a lot of, I didn't bring a lot of introductory slides, assuming that versed on leishmaniasis. So I'm going to start from the clinical outcomes immediately, but just in case, it's a vector-borne infectious disease, okay? So what I wanted to show with this slide, if you allow me, I prefer this side. This side. What I wanted to show with this slide is that infection with Leishmania has a range of different clinical manifestations. This is true for visceral leishmaniasis as well as for cutaneous leishmaniasis. In the context of cutaneous leishmaniasis, we have a high proportion of individuals that upon infection will never develop symptoms, and this is the group of people that we call asymptomatics. And then among the individuals that have symptomatic infection, we have also a range of disease severity that can go from very mild self-healing lesions to very chronic, highly inflamed with tissue destruction, and these are chronic manifestations of cutaneous leishmaniasis. Among the chronic manifestations of dermal leishmaniasis is mucosal disease, which I think you are all very well acquainted with, as Brazil is the main, one of the main countries where we have the highest cases of mucosal disease. But the other important thing from this slide is that we always have to keep in mind that leishmaniasis is the disease itself is an immunopathology. It's not like a bacteria that causes disease because it secretes a toxin and the toxin degrades tissue or promotes tissue destruction. Here is the immune response that induces finally the destruction of the tissue. Parasite elicited, but immune response mediated. We have, and this is, I cannot say how happy I am to be in a place that is devoted to vaccine investigations for all of these infectious diseases of poverty, especially cutaneous leishmaniasis, Chagas disease, because as you are aware, of course, there is an urgent need for a vaccine for our infectious diseases. And this is in addition, not only because we have a high number of cases, but also because the therapeutic strategies are very limited, are extremely toxic, are very difficult to get to the communities because they're either parentally administered or they have high toxic profiles that need to be taken care of during the course of treatment. And so it is imperative that we have treatment strategies that are applicable in the communities where disease happens, and also hopefully disease-preventing vaccines. Recently, in the past 8 years or 10 years or so, we have been lucky enough that finally WHO and PAHO agreed to approve the use of local treatments for cutaneous leishmaniasis, but this is something that starts to give us an idea of the issue that we're going to be discussing here, which is persistent infection. I don't know if you know, but local therapies were not approved in the past for treatment of Leishmania aviana infections because of the concern that there was going to be development of mucosal disease. Because if you treat locally and you don't treat systemically, then the bug is not going to go from the body, and therefore you have the risk of development of mucosal disease. Then many years after, a lot of research came to show that even if you have clinical cure of disease, you still have parasite persistence. So there is no argument whatsoever for the use of systemic disease, of systemic drugs, unless you have, of course, very complicated manifestations. But this argument of parasite persistence and risk of development of mucosal leishmaniasis is still there, even if you treat systemically. So that's why local treatments were approved, although not for all cases of cutaneous leishmaniasis, and that's something that we can talk about later on. And moving from this issue of parasite persistence, the first thing that I wanted to do was that we have clarity in the room of what is it that we're calling parasite persistence. So I'm not here talking about persister parasites. I think a lot of you have a lot of understanding of the molecular mechanisms of the persister parasites that are metabolically inactive and therefore, dormancy, quiescence, etc. What we're talking about here is persistent infection, okay? We don't know how the parasite is, but we know that the parasite is there. So when we talk about persistent infection, what are we talking about? We're talking about individuals with asymptomatic infection that you test tissues like the mucosa, the nasal mucosa, the tonsils, or the conjunctiva, for example, and you can find molecular evidence of presence of the parasite there. Those are individuals with persistent infection. We have individuals that have had symptomatic disease that after you provide treatment and they cure, they also have persistent parasite infection. Again, in these individuals that have cured, in a proportion of them, if you take samples from healthy skin, from the nasal mucosa, conjunctiva, for example, you're able to detect the parasite there. And the other type of persistent infection that we talk about is the issue of treatment failure. And this is one of the most interesting in the sense that you have still tissue destruction, but there's almost no parasites in there. So what's going on? So today I'm going to tell you 1.5 stories. The one story is about treatment failure, and the half story is about individuals that have healed. So we're going to be seeing the phenomenon of parasite persistence in these two different contexts. In clinically cured but parasitologically persistent individuals, this one, and in parasitologically persistent individuals with clinical manifestations of disease. So let's start here. So this is what we imagine when we hope to get a clinical healing of a Leishmania wound. Essentially what we have is that we have the parasite that inoculates spermastigotes. This elicits immediately, because of the salivary proteins, but as well the parasite itself, it stimulates a very strong recruitment of pro-inflammatory cells. There is immediate production of pro-inflammatory mediators, and the idea is that you get phagocytes, including neutrophils, macrophages, dendritic cells, to phagocytize the parasite and hopefully eliminate the parasite from these sites. However, when that is not very efficacious or efficient, what happens is that the parasite establishes an appropriate niche where the parasite can actually propagate intracellularly. It transforms into a mastigotes. These mastigotes go again and replicate and promote even more recruitment of inflammatory cells, which begin the process of tissue destruction, which at the end of the day we see as the leishmaniasis lesion. And essentially what we're hoping to see when we give a drug or when a person has self-healing lesions, for example, is that this inflammation or this inflammatory environment is eliminated, let's say it's controlled, that hopefully the parasite is eliminated, and then you have restoration of the epithelia and reepithelization of the lesion. However, what we see in the cases of treatment failure in cutaneous leishmaniasis is that we still have a very strong inflammatory environment, and then we have parasites, but parasites that are a very, very, very low parasite burden, almost undetectable. And I would say that in about 80% of the cases of treatment failure, if you go and you take a sample from the lesion, you're not going to be able to detect the parasites in those lesions that have failed. Even through PCR, yeah, it's incredible. So the parasite burden is really, really low. So what's going on here? We decided to tackle this situation because our hypothesis was that there was a deregulated immune response that was responsible for that treatment failure. It was not necessarily that the parasite was drug resistant or that the parasite was in a very high parasite burden in those tissues, but that indeed there was a deficiency, let's say, in immune regulation that avoided tissue healing. So this was a project that we finished about 2 years ago, and essentially we did recruitment of patients with cutaneous leishmaniasis from 2018 to 2022, and essentially what we did was that we recruited patients with active cutaneous leishmaniasis, and the idea was that we were going to take samples of the skin or the lesion and blood samples to analyze the innate immune component. And we were interested in the innate immune component is because we have seen in previous work that the innate immune response seems to be the primary driver of this pro-inflammatory response that you see in those non-healing lesions. And so what we designed was to take samples pre-treatment, midway through treatment, and at the end of treatment to see how the dynamics of inflammation would change over the course of treatment, both in the tissue, but as well systemically. And this is important because we always think that what we're going to see in the skin is going to be mimicked to what we see in the blood, or on the contrary, the blood response will mimic what is seen in the tissue. And this is what we wanted to evaluate here, and you will see how this is not necessarily the case. So yeah, this was all patients, just so that you know, were treated with antimonials. Some were treated with miltefosine, but most of the data that I'm going to show you is with antimonials. All right, so the work that we did here was a very thorough transcriptomic profiling of the, I forgot to say, of course, but this is kind of self-evident. Monocytes, neutrophils, and eosinophils. These are the cells that we were evaluating. And the idea was to do transcriptomics of all these samples. So this is the first main result that I wanted to show you, but this is a simple PCA where we see in different colors, different samples. So we have here biopsies, we have here neutrophils, monocytes, and eosinophils. And what's important to see in this slide, yes, of course? No. So thanks for the question, because this was a lot of work you cannot imagine. So this was isolation of monocytes, neutrophils, and eosinophils. And then from the isolated cell population, we did the bulk transcriptomics.
So it's an issue with some collagenase and...
No, so for the biopsies, no. Okay, so let's come back here. From blood, we isolated monocytes, neutrophils, and eosinophils from blood. From the skin, it was the whole biopsy that we used for bulk RNA. This? No. These were all done by max purification. So we don't have a FACS sorter at home, so we do it by how do you call it, magnetic beads. Mm-hmm.
The use of fuel population. Do you have any?
We have a what?
Any increase of the use of fuel disease?
That's a great question. You're the only person that have asked this before. Yes. So this is incredible. So we can talk about that later. But no one checks the hemogram of the blood counts in patients with cutaneous leishmaniasis, because this is a localized disease. What we did with a study of, I don't know, it was maybe about 300 patients. Unfortunately, we have not published this, but about 300 patients that have gone into all our clinical trials. We take always a blood cell count. All the patients with cutaneous, not all, but the majority of the patients with cutaneous leishmaniasis have eosinophilia. The majority. But it's not a very high eosinophilia, but there is eosinophilia.
It's very interesting because in human, visceral leishmaniasis is the opposite.
Okay.
There is eosinophilia.
Okay.
And nobody cares about eosinophilia because it just says like a little number, but basically it disappears. But then you say, is this because of the leukopenia?
Exactly.
But in your case, it's...
Exactly. And the other thing is that they are very annoying to work with. And so everyone just goes like, no, let's just go to monocytes. Exactly. Yeah. Thanks for that question. Yeah. All right. So the question that you ask is very important, is because we all know that when we do transcriptomics of neutrophils, we have to be very careful with the monocyte contamination. And so all the neutrophil preparations were always checked for FACS so that we don't have monocyte contamination. And if there was monocyte contamination, it was allowed to be maximum 2%. So this was a good first result, that tells us that our transcriptomes are pretty cell-specific or sample-specific. So I'm going to start first with the biopsies. The biopsy work, so we were hoping, or our expectation was that the biopsies were going to be revealing, that they're going to give us the best biomarkers ever. And you may have read a paper that was published by Edgar Carvalho and his group with Phil Scott. And they did some transcriptomic profiling of lesion biopsies as well during treatment of patients with Leishmania brasiliensis. And they showed something related to cytotoxicity in the lesions. We also saw the same cytotoxicity, but what I want to show you here is something different. When you see the PCA profile of the biopsies, what you see very clearly is that the pretreatment samples cluster almost independently to the ones from end of treatment or mid-treatment. And this is important because this starts to give you an idea where the main differences are going to be observed. It's not necessarily at the end of treatment where we're going to be seeing the main differences. And this is observed as well when we look at the volcano plots. So what we have here are volcano plots of the differential expression analysis of pre-treatment against mid-treatment, pre-treatment against end of treatment, and mid-treatment against end of treatment, in patients that cured and in patients that had treatment failure. If you see, when you compare the pre-treatment samples compared to the mid-treatment samples, there is an evident increase in the number of genes that are differentially expressed in patients that cure compared to patients that have treatment failure. So here we have a total of about 300 genes, and here we have a total of about 800 genes. And when you compare the end of treatment versus mid of treatment, in patients that cure, there's almost no differential expression. And this is surprising because how is it possible that there is not a big change in the transcriptomic profile in the lesion midway through treatment towards end of treatment? And what I want to show you here is something that is even more strange. These are pictures, sequential pictures obtained for a single patient. So a column is one patient, and what we have is pre-treatment, mid-treatment, end of treatment. What I want you to pay attention is this mid-treatment. These lesions, independent if the patient has cured or progresses to treatment failure, most of these lesions are still very active. They are pretty, active. But then you go back to here, and there is essentially nothing happening when you contrast mid-treatment versus end of treatment. So how is this possible? So essentially, I'm not going to show you all the data of the network analysis, but what this work told us was that the main transcriptional profiles that are associated with a healing response in the lesion are established very early upon treatment. They are established during the first 10 days of treatment. And this is important to keep in mind in cases where you want to think about immunomodulation. Because if your healing response is established early on, if you give an immunomodulator later on, for example, you're not going to have an environment that allows for proper healing. And so this we published last year, but this is a nice picture that summarizes those results. So essentially, what we see is that when we compare pre-treatment against mid-treatment in patients that cure, we have a very strong downregulation of the pro-inflammatory response that is mediated by IL-1, by interferon signaling, primarily type 1 interference, and also the gene expression profiles that are associated with neutrophil activation and degranulation. Simultaneously, while this is decreasing the inflammatory response, simultaneously we have an upregulation of the extracellular matrix reorganization and essentially tissue proliferation. So we have collagen formation, extracellular matrix organization, and genes involved in keratinization. In contrast, what we see in patients that have treatment failure is that we have a decrease in genes associated with keratinization and cell cycle progression and cell replication. So essentially we have a lot of necrosis there and not replenishing with new cells. And we have activation of the immune response mediated by complement activation and Fc gamma receptors. So just to summarize this part, this gives us insight into the moment where it would be ideal to have add-on therapies, which would be between day 0 and day 10 when you treat with antimonials. And then what about the parasite load? So we're usually, again, we're microbiologists, we're always thinking, oh, you kill the parasite, you heal the patient. And so essentially the same is true for leishmaniasis, although this is not necessarily the case in these patients. So what we see in terms of parasite load is that independent if your final outcome is cure or treatment failure, you have an almost undetectable parasite load at the end of treatment in these patients. And so that means that the persistence of the lesion is mediated by the inflammatory response and not necessarily by the parasite that is persisting there. And this was a study that was done by Nancy and a colleague of ours that evaluated the time that it takes for the drug to start killing most of the parasites from the lesions. And essentially what they did is that they recruited patients, took lesion swabs of the cutaneous lesion before treatment and at day 2 or 3 or at day 8 after initiation of treatment. And what you can see here is that the reduction of parasite burden is very strong very early on after initiation of treatment. So those lesions have a very reduced parasite burden very rapidly. And this is, that was not known, this is very new, but it is known that the antimonials get to the skin very quickly. So it makes sense that something like this is occurring. And then by day 8, it's almost undetectable. Now, we don't know, and I think what happens is that once you stop giving the antimonials, there are some parasites that persist there, and those are the ones that most likely become quiescent or metabolically inactive or persisters, if you want to call it like that. But this is a very clear evidence that there's something in the immune response that is not being taken care of and that maintains these lesions. And so that's when we looked at the innate cells from these patients. So essentially what I'm going to show you is the other compartment of the story. Can you please tell me when I have 5 minutes left? Thanks. And the first thing was, we saw in lesions that there was a difference in the transcriptomes before initiation of treatment against mid and end of treatment. And so the same thing we were going to analyze for monocytes, neutrophils, and eosinophils. And what you can see very clearly in these PCA plots is that there is no separation whatsoever of the transcriptomes. They're like the transcriptomes that you take before treatment are very similar to those midway through treatment and very similar to those at the end of treatment. This is important in the context of the discussions that we were having before. These are cells that have not been re-stimulated with anything. These are cells straight from the blood, straight to the sequencer, essentially. Well, straight to trisol and then to the sequencer, but they are untouched. And and even if you include the visit number in your differential expression model, the analytical model for differential expression, essentially is the same data set that you get with visit in the model or without visit in the model. These are just correlation plots where we have visit in the model and visit not in the model, and essentially we have the same gene expression profiles. So what we decided to do here is, okay, if the visits, if there is not a huge difference between the visits, let's pile together all the samples from all the cures and all the failures, and let's look what this can tell us. And this is it. Essentially, when we do the PCA plots using SVA with monocytes, eosinophils, and neutrophils, we can see a very clear discrimination between the transcriptomes of patients that have treatment failure versus those that have a cure. And this was true for monocytes, eosinophils, and neutrophils. And this was amazing. And what is more amazing on top of this is that when you look at the differential expression profiles, they were almost identical. Not in the genes, because the genes are different, but in the functional signatures that they were reflecting. So essentially what we have in all the different innate cells was an increase in type 1 interferon response genes in treatment failure patients. So that means that there is this type 1 interferon signaling that may be associated with treatment failure. even when you take all the different samples, neutrophils, monocytes, eosinophils, you put them together and you do differential expression analysis of cures versus treatment failure, same thing you find. We have increase in type 1 interferon responses, and this is what we are hypothesizing that is mediating tissue destruction. So I told you that the commonalities were not necessarily the genes, but were more the functional signatures. GO. So if you compare the genes, only 12 genes are common to the 3 differential expression analyses. But among these 12 genes, most of them are associated with type 1 interferon responses. So what we did with this study was we took the expression, the RPKM data for all these genes, and we constructed a composite score aiming to identify or to create a predictive signature of treatment failure. And essentially what I'm showing you here is the performance of that score when using only the pretreatment samples. So we don't think that we're going to be able to do a transcriptomic profiling by any means as a predictive, clinically applicable thing for leishmaniasis, but this gives us some insights into what routes we can start exploring. Okay, and then what starts bringing us to the next part of the story is, again, the issue of the parasite. So what I've shown you so far is that there is this hyperinflammatory response that is occurring in patients with treatment failure, but what happens with the parasite? So what's really cool is that we can look at all those transcriptomes from the parasite point of view. So we can do the analysis annotating to the parasite genome. And essentially what I'm showing you here is the number of splice leader reads that are detected in the transcriptomes of monocytes, neutrophils, eosinophils, and in the biopsies in both patients that cure and patients that failed. And our expectation was that we would find more in our failures, less than our cures, but this is all identical. But what's I still don't understand, and it's very interesting, is this thing. You're finding parasites in cells that are very short-lived, in neutrophils and eosinophils. And if you look at the correlation of the parasite loads based on SL reads between monocytes and neutrophils, there is a positive correlation, meaning that those patients that have higher numbers of parasites in monocytes have also higher numbers of parasites in the neutrophils. So what's going on here? We don't know, but I think that this is a very interesting thing to continue exploring. But it doesn't matter if it's cure or failure. It doesn't matter if it's cure or failure. It doesn't matter at all. So it's not a parasite. But no, but this is a big discussion that we have back at home. Anyway, I think it's not a parasite. And so if we're seeing our parasites appearing in these different cells, and this is in the circulation, where is the parasite persisting really? So we know that it's in the blood, we know that it's in some phagocytes that we can find in the blood, but then I mentioned to you at the beginning of the talk that we find also parasites in different mucosal tissues. So if you recall, in leishmaniasis caused by Leishmania aviana, the parasites are able to migrate to mucosal tissues, and that's why you have mucosal leishmaniasis. And so Nancy did a study in 2009, if I recall correctly, where they took samples from the nasal mucosa, the conjunctiva, and the tonsils of patients that have healed, or they just have a cutaneous lesion, but no clinical manifestation whatsoever of disease in the mucosa. And what they found was that you can detect the parasite in this nasopharyngeal mucosa of patients that have either cutaneous lesions or that have asymptomatic infection or healed. And what's interesting is that the most informative samples to find these parasites are blood, as I showed you before, but also the nasal mucosa. And so we became interested in this nasal mucosa as a site for parasite persistence. And I bet you, for those of you that work with Chagas, I don't know why no one has done this before, but I bet you, like almost hand in fire, that you will find the parasites in the nasal mucosa as well. You will see. I'm sure. I mean, but anyway. This is just for anyone that works with patients. Anyway, so what we did, and this is the project that is ongoing now, is we became interested in these different tissues that may harbor persistent parasites, but interested in those patients that have healed. And this is the half the story that I mentioned at the beginning. And why half is because we're still doing the recruitment of the study cohort. But essentially what we're taking from these individuals are the same type of samples that I mentioned before, in addition to nasal mucosal samples. And so what I brought here, this is data that literally came out maybe 2 or 3 weeks ago. So this is very, very recent, very preliminary, but I wanted to share it with you because it may be useful for you guys as well. We take two approaches. One, we take nasal curettes. That's like the microbiological, la essa de microbiología, the thing for bacteria culture. Essentially, it's something similar, and you take just a scratch of the nasal mucosa. Or we take a nasal swab. And these are some pictures of the types of cells that you will find in nasal mucosa, the epithelial cells, but also we find immune cells such as neutrophils, monocytes, CD4, CD8 T cells, etc., etc. And then when we do the transcriptomic profiles, this is another cohort of patients. And again, what we have here are samples from scar tissue and from healthy skin, PBMCs, white blood cells, and these are the nasal mucosal samples. When we look at the nasal mucosa and we start analyzing the differences between individuals that have, all of them, remember, have healed. So these are people just like you and me. We take a sample from the nasal mucosa and we test by PCR whether we detect Leishmania in those. So what you see here is in green, samples from nasal mucosa of parasites that are PCR negative, and in orange, samples from patients that are PCR positive in the nasal mucosa, but with no symptoms of disease at all. So we start seeing some discrimination. It's not great. The variance is small. But even so, what happens if we do the differential expression analysis? And what's very peculiar is that we start seeing a downregulation of genes involved in MHC class II presentation and antigen processing. So this is the GO analysis, essentially a repeat of what I just mentioned. But what this starts telling us is that there may be a permissive environment in the nasal mucosa that is mediated by dampening of antigen presentation. So these are just the first set of results that we're getting. And then again about the parasite, what happens with the parasite? So what I'm showing here is a set of pairs of strains. This is very, very unique set of strains that we collect before. So a patient arrives to us, they have a primary lesion. And then they come 1 year or 2 years later with a secondary lesion. So this is pairs of patients, pairs of strains obtained from the patient at the first case of cutaneous leishmaniasis and then at recurrence. And what you can see very clearly is that the pairs of patients cluster together. The number is the strain, I for initial, R for recurrent. 103, 103, 114, 114, and so on. The only one that is different is this 112, which is actually, we know that this is a reinfection instead of a reactivation because they are different cymodems. So essentially what I want to show you here is that genetically speaking, because these are SNP profiles, the parasites are identical, the persistent recurrent versus the initial one. And then this is just, I think, my final slide, just to show you some of the things that we're trying to do in the lab, because, you know, working with patients is a little complicated. And so what we're trying to do is to model systems where we can recreate persistent infection. And to do that, we are working with a 3D cell culture system using spheroids that are generated with collagen and alginate to try and model the skin tissue. And essentially what we have been able to obtain here are cultures. I don't have the final number here, but these are 3D cell cultures that have cell viability up to 15 days. So please bear with me. They are viable, very viable at 15 days. And we also have the parasite where we can detect it at 15 days. Of course, you have a decrease in the parasite load, but we still have persistent parasites. And our hope is that we can use this system to inquire a little bit more about the parasite and the mechanisms that the parasite is using for persistence. So in summary, I hope I have shared with you that disease is not linearly correlated with parasite load, and this is important when we're thinking about drugs. That the inflammatory response is mediated by activated monocytes and neutrophils, which sustain pathology, and this is related to what I mentioned before about the CD8 cytotoxic response. So what we think is that the systemic neutrophil and pro-inflammatory response that we're seeing is triggering the CD8 T cell response that is observed in the lesions. That Leishmania persistence is the norm rather than the exception, and anyway. That parasites are predominantly, they persist outside of the skin. I didn't show you this data, but finding the parasite in the skin is very, very difficult. We find it more frequently in the mucosa. And the transcriptional profiles of nasal mucosa can differentiate tissues that favor persistence. This is tentative still, but this is what we're starting to observe. So with that, I close. I thank, of course, all my group, all the clinical team that has helped us, or all our community leaders with whom we would be nothing. My friend and colleague Najib El Sayed and Trey, who are the people that work with us in all the transcriptomic analysis, and our funding that comes from Wellcome and NIH. And before I finish, just give me just 2 seconds, because you're a vaccine group. I wanted to tell you a little bit about this. This is a network that is called Validate. This is a network of investigators that work in vaccine development and investigation in 4 different pathogens. In mycobacteria causing leprosy and tuberculosis, Burkholderia causing melioidosis, and leishmaniasis. So for you guys that are part of the work with Leishmania vaccines, I encourage you very much to participate in this network, because this network has a lot of advantages. You can get funding, you can get funding to go and travel, to learn techniques to different labs. You have a cohort of group of people that work with the same problems that you work, and we're starting to create regional subcommittees. So I am in charge of the regional subcommittee for the Americas. So I'm encouraging all of you to participate. I'm also co-director of the network, so in a way this is a good pitch. But essentially you have a lot of interesting things. So you can join as an investigator, as an associate or affiliate, depending on your career stage. But for example, if you're an associate, meaning an early career investigator, for example, you can have the opportunity to get fellowships for 2-year, for conducting your own project for 2 years, pump priming grants, travel grants, etc. It's a really cool network. And then I go back to this in case you have any questions.
The money comes from Gates Foundation?
Gates and MRC.
Gates and MRC, yeah.
But that's for Validate. My money for my research,
Yeah.
Wellcome and NIH.
Oh, no, yeah. For the
For it, yeah.
we have questions. Is that working here? Yeah.
Great talk, Adelaide, and it's very nice to see the other side of the picture for Leishmania. My question is in terms of you have the opportunity or have gene expression in 3 different time in terms of the outcome after you start treating. What's the profile of gene expression of the parasite in the beginning of treatment in terms of there is change in the repertoire of gene expression by the Leishmania?
Great. So that's a great question. Let me see if I have, I'll reply and then I'll see if I have a slide to show you. So we, together with Najib as well, we did a huge transcriptomic profile of about 120 different strains isolated from patients that have a prospective cure or failure. There is no association whatsoever of the transcriptome between patients that fail and patients that cure. But where there is a difference is in the transcriptome profiles between drug-resistant and drug-susceptible parasites. Now, this is a tricky mix because drug resistance is not always associated with failure. So, transcriptomes differentiate resistance and susceptibility, but not failure and response. Now, in terms of kinetics very difficult. We cannot, what we see, so what I showed you here, what I showed you in the, for example, with the SL reads. So those SL reads come from pre, mid, and end of treatment. When we tried to look at the differential expression profile, it was extremely difficult because the amount of reads for coding regions was very low. So the coverage was minimal. So we have to rely on either pairs of strains, as we're doing here, or in the primary isolate, comparing between cures and failures.
Hi, Adelaide. Great to meet you. Great to listen to your lecture. My name is Greg. I'm a veterinary medicine doctor, and I worked with dogs a lot of my life. And it was very intriguing, the number of dogs that we found when you do the blood counting and you find neutrophils infected with Leishmania. Was very astonishing. It's very common. And a few years ago, I just by chance, I began a project studying leishmaniasis in a mouse model that do not have eosinophils. But they also have some off-target problems. But we found that these mice, they cannot control the disease, and they usually metastatized before the wild type. It means that by 9 weeks, they have much more metastatic lesions. I used Leishmania amazonensis and also Leishmania major. And we conclude that eosinophils are somehow controlling the early dissemination, which is kind of the opposite that you found, because you have, of course, you didn't. It wasn't a hypothesis, but you have a lot of cells in the blood carrying the parasite, and this was actually actually a mystery how the parasite disseminates through the body. And I would like to hear a little bit more on your thoughts about that, and what you think about this several blood types, cell blood types carrying the parasite to another organs.
I'm convinced that that's the way. So let's go. The eosinopenic or eosinodeficient mice. So I don't think that is necessarily contradictory. So what happens in, or what I interpret from our findings, is that the problem is the sustained inflammation. So I'm sure that we always need a good pro-inflammatory response at the beginning to get rid of the parasites for tissue healing. The problem is that this regulatory response is not arriving well, and that is what leads to treatment failure. This is the hypothesis that we have, and indeed there are some studies that show that Tregs in these patients are not functional. They're very abundant, but they do not produce interferon gamma, IL-10, etc. Yeah. Now, in terms of the cells that harbor the parasites. So I am very intrigued by the neutrophils and the eosinophils because they're short-lived cells, right? But if you go and you really go deep in the literature, very, very deep, you find these publications that show that neutrophils can be a little bit longer-lived than the 3 days that we always think. Now, what I think is not necessarily that they live forever, but that there is a cycle of phagocytosis of the neutrophils that sustains the parasite there.
Enough for them to go and disseminate.
And to persist in the neutrophil population as well.
You also have, if you go deeper in the literature on the neutrophils, you'll find that there are some neutrophils that live a little bit longer. And they have a very different pattern of expression than the other that they are short-lived.
Exactly.
And the other question was actually, but I'm not sure, have you do any single-cell sequencing of this parasite?
No.
Because I think a next step is probably to do a single-cell sequencing in these parasites in these cells on the blood, if you to check whether the the cells carrying the parasites have a different pattern expression that
Totally.
might explain.
No doubt. Yeah, totally.
Thank you.
Sure.
Anybody? Another question? I have one quick question just to add a little bit more confusion to the whole scheme. What about the Leishmania RNA virus? Can you use your data to find these LRVs? Because
So we tested. This is the first thing that comes, okay? We tested all of our patients. All the data that I've shown you here is LRV negative. Now, adding to that, we have traced maybe 120 strains. Leishmania, I forgot to mention this. I work with Leishmania panamensis. Exactly. So in our Leishmania panamensis strains, which are the most cases here, the virus, we cannot find it. We cannot find it. There are some people in Panama that have found it in some strains in Panama, but in our strains from the Pacific coast, we don't find it. We find it in Brasiliensis and in Guyanensis. So treatment failure in our case and pathology is not mediated by the virus. Maybe another one. Well, yeah, I have this. Yeah, he knows.
So, anybody else have a question? If not, I would like to thank you very, very much indeed. And then we're going to stay here for a little bit more discussion. Anybody wants to keep talking to Adelaida before she leaves, will be nice. So we can stay here in the auditorium. And no questions in the chat. And I thank you all, and thank you again.
Thank you all.
Bye-bye.
Happy to be here.